Embryo quality could be evaluated according to morphology and according to timing of the cell division. Morphology grading system is based on especially number of cells, so that means like 2 cells, 3 cells, 5 cells, 8 cells. Then we can evaluate stage specificity, which means that the specific volume of the or dimension of the cells blastomere within the embryo is specific for each cell division. So we can recognize if the division till 5 cell stage embryo, for example, was done properly or not. Then often is evaluated the number of the fragments, cell fragments, which could be visible during the cell divisions. And then there is some another marks which could be visible, and according to that we can evaluate the embryo quality as well, like multinucleation in blastomeres, cell morphology, granularity of the cytoplasm within the blastomeres, and other marks like this. This grading system is normally used till the embryo is growing till day 3. On day 4, so then the compactation of the cells occurs. So then we evaluate the compactation if it's done within whole the embryo, so like fully compacted morula, for example, or if the compactation occurs just within the small part of the cells or bigger part of the cells, so then that's a partially compacted morula. On day 5, when the embryos reach the blastocyst, so we usually use the grading system based on blastocyst expansion and hatching status. Then we will grade the inner cell mass, and then we have to grade the quality of the trofectorare. The expansion of the blastocyst could be evaluated according to cavity inside of the embryo, which could be small, full, or expanded, or the blastocyst already can go out of the zona pellucida, which is called hatching, or the blastocyst could be actually out of the zona pellucida, and that's the last stage of the blastocyst expansion, and this is fully hatched blastocyst. Inner cell mass, that's the group of the cells which create embryo itself. We evaluate how many cells create inner cell mass and if they are compacted or not. The same grading is used for the evaluation in trofectorarium, which create placenta later on. And we again, in that group of the cells, evaluate how many cells create trofectorarium and if they are tightly grouped or loosely grouped. If we need to evaluate the embryo according to timing of the cell division, so we need to use a special incubator which is called time-lapse incubator, and using this we can take a picture during whole development of the embryos, during the whole culture, and then we can see when the cell division is done. We use the software which help us to evaluate all these timings, and then we would say which embryo has a higher potential to give the pregnancy at the end after the transfer.